Producing a pharmaceutical drug is an exercise in absolute precision. A manufacturer must guarantee that the millionth dose administered to a patient is exactly as safe, pure, and effective as the very first. For decades, the industry attempted to prove consistency using a standard known as the 3-batch rule. If a facility produced 3 consecutive successful runs without failing, the process was officially declared validated. This graph illustrates the statistical flaw. Relying entirely on 3 isolated events, these green check marks, fails to measure continuous capability. A highly variable process can fluctuate wildly, yet by pure statistical luck, hit passing thresholds exactly when sampled, masking an unstable system. Because of this mathematical blind spot, global regulatory bodies now reject this outdated heuristic. The Parenteral Drug Association's Technical Report Number 60 explicitly states that isolated successes do not constitute a state of control. Process validation is no longer treated as a one-time event, ending after a few good runs. It operates as an ongoing, continuous life cycle of evidence collection. True validation requires establishing documented, statistical proof that a process is inherently designed and capable of delivering quality product over its entire commercial lifespan. This life cycle begins with Stage 1, process design. This phase occurs entirely during early development and scale-up, before commercial manufacturing even begins. Engineers start by defining critical material attributes, or CMAs. These are the inherent physical and chemical properties of the raw input materials used in the drug. Next, they identify the critical process parameters, or CPPs. These are the specific processing dials, like temperature, pressure, or blending speeds, that operators can directly control on the equipment. The goal is to determine exactly how those raw material inputs and equipment settings interact to produce the final critical quality attributes, or CQAs. CQAs are the defining biological and chemical characteristics that make the finished drug safe for a human patient. This 3D topographical grid visualizes that complex, interconnected relationship. By adjusting these sliders for material inputs and dials for process parameters, scientists establish a multidimensional operating window known as the design space. If any of those dials are pushed past the established boundary line, the resulting product immediately falls out of specification and fails. Rigorously mapping the precise edges of this multidimensional design space is the singular objective of Stage 1. This methodology is anchored in the principle of quality by design. Quality by design asserts that safety must be engineered directly into a product, because you cannot validate a mechanism you do not comprehensively understand. Stage 1 defines the exact boundaries of what a safe state of control looks like, replacing floor-level guesswork with a predictive scientific model. Once the boundaries are modeled, the life cycle moves to Stage 2, process qualification. This phase proves whether the theoretical strategy actually functions inside a physical good manufacturing practice, or GMP, facility. This begins with facility and equipment qualification, a prerequisite step ensuring the physical hardware and utilities operate as intended. But the crucial bottleneck of this phase is Stage 2B, process performance qualification, known across the industry as PPQ. A successful PPQ campaign serves as the absolute regulatory gatekeeper. Without it, a new therapy cannot be commercially distributed. PPQ is an exercise in advanced statistical confidence. It is designed to prove the process performs consistently under normal commercial conditions. This control chart shows how modern sampling strategies operate. The required number of PPQ batches is not an arbitrary fixed number. Instead, a wider distribution curve, representing higher residual risk remaining from Stage 1, automatically forces the execution of a more intensive, dense sampling plan. As the facility generates that dense sampling data, the curve narrows and centers. The process proves it can anchor itself safely and rigidly within the control limits established back in Stage 1. This exact mathematical certainty replaces the blind hope of hoping 3 arbitrary batches happen to pass. A robust PPQ delivers irrefutable proof that the control strategy works at a commercial scale. However, approval of the PPQ does not mean the validation is complete. Production enters Stage 3, continued process verification, or CPV. This is the relentless, ongoing maintenance of the validated state over years of routine commercial manufacturing. The physical reality of manufacturing is that processes drift. Over thousands of hours, microscopic equipment wear, subtle environmental shifts, and minor variations in raw materials pull a process away from its ideal center. This shoe-heart control chart illustrates how CPV tracks that invisible drift. A shoe-heart chart is a statistical tool that plots process data over time against defined limits. CPV uses it to monitor real-time commercial batches as they leave the line. The scenario engineers are trying to prevent is an out-of-specification, or OOS, failure. An OOS result occurs when a batch completely breaches the absolute upper control limit and fails its acceptance criteria. An OOS result is a severe quality failure. It represents millions of dollars in wasted materials and directly threatens patient supply by causing drug shortages. To prevent this, CPV relies on the out-of-trend, or OOT, signal. As the data points slowly drift upward and cross an internal statistical warning limit, it triggers a yellow alarm long before the absolute failure line is reached. An OOS is a reactive failure. An OOT is a preventative early warning signal. When an OOT signal fires, engineers can intervene immediately, recalibrate the process parameters, and pull the system back to the center before a single batch is ruined. Engineers feed this CPV data directly back into the development loop to drive continuous process optimization. Continuous monitoring replaces reactive crisis management with a proactive, self-correcting system. This blueprint visualizes the complete framework in PDA Technical Report 60. The 3 phases function as a unified, continuous, closed-loop engine. Stage 3 routes real-world data back to Stage 1 to optimize design, and to Stage 2 to trigger requalification. Stage 1 sets the boundaries through rigorous scientific design and risk assessment. Stage 2 proves that design is viable at commercial scale using strict statistical metrics. Stage 3 monitors the physical reality on the floor continuously to catch and correct drift. For engineers and quality assurance personnel, process validation can no longer be viewed as a rigid regulatory checkbox required to start production. Embracing this continuous life cycle aligns business efficiency with strict FDA compliance. A controlled process yields fewer failed batches, minimizes material waste, and ensures predictable output. Ultimately, this framework provides the mathematical and scientific rigor required to fulfill the industry's highest mandate, guaranteeing uncompromised consistency and safety for every patient.